Good morning all, good morning Sadhguru, good morning Lakshmi, good morning all. Good morning Shivani, good morning Reen, so yeah. Good morning all. Good morning Priya, good morning Priya. I think Tarun you missed yesterday class also, no? Whomever missed the classes, please kindly watch the recorded, don't miss the classes at all. Okay, perfect. No problem. Okay. See friends, please be regular. When we are regular and when we are consistent, then only we will succeed. Okay. Okay, that's good. So friends, in the last class what we discussed? In the last class, we have completed the hormone auxin and even its signaling also. So friends, today we will try to finish the 2 to 3 hormones. Good morning Saima, good morning Jayashree. Please continue. Okay, so friends, listen to me for the period of 10 to 15 minutes. After 10 to 15 minutes, I will solve all the doubts. And friends, listen to me when you are reading the hormones, as I told you in the last class, the best way of remembering the hormones is comparing one hormone with the another hormone. When you compare one hormone with the another hormone, you can easily remember. Good morning, Deepika. Continue. So friends, listen to me everyone. The discovery of the gibberellic acid is really funny and accidental actually. So all the students listen to me very carefully friends. Originally, what is the height of the rice seedling friends? Okay, Tarun, that's good. Okay, please continue. What friends, in the Japan, the village farmers, they observed certain rice seedling which has grown up to 13 to 14 feet. Up to 13 to 14 feet. Originally friends, the rice seedling will grow up to how many feet? Originally the rice seedling will grow up to 3 to 4 feet tall. But my friends, they have observed that certain rice seedlings are growing up to 13 to 14 feet tall. And in the village language, all the farmers thought that maybe because of a certain diseased condition, the plant is growing like this. You know what the farmers does? In their own village language in the Japan, in their village language, they have given the name Bakane disease. Friends, focus, which means foolish seedling. In other words, which is growing unnecessarily. Bakane disease or foolish seedling means which is growing unnecessarily. But friends, later an intelligent scientist named as Kurosawa, everyone listen friends, Kurosawa, he identified that this excess growth is not happening naturally. This excess growth can be happened because of a particular chemical. And friends, who is going to release that particular chemical? That particular chemical released by the Gibberella fusicori. So this is the name of the fungus. Whenever this fungus attack that particular plant, this fungus is basically releasing a chemical. That chemical only, my friends, we will be calling it as the gibberellic acid. Okay, everyone very quickly recall. The discovery of the gibberellic acid was really funny and accidental. In the Japan, the village farmers observed that some of the rice seedling grown up to 13 to 14 feet long. Originally, my friends, this particular rice seedlings cannot grow that much height. They will hardly grow 3 to 4 or maximum 4.5 feet. Later, the farmers thought that because of a certain disease, it is growing like this, and they have given the name to that particular disease is what friends? Bakane disease or foolish seedling. Please kindly don't forget the statement at all. Later what they understood, later a scientist said that this particular growth is caused by a chemical secreted by the fungus. What is that particular fungus, friends? We can call it Gibberella fusicori, and name of the chemical is Gibberellin. Friends, every one of you understand initial time. In friends, you have to remember most of the information. Initially, friends, three Gibberellins were isolated. What are the three Gibberellins that were isolated, friends? One is the GA1, one is the GA2, and one is the ZA3. These are all the three Gibberellins that were identified initially. But my friends, every one of you listen, so far there are more than 150 types of gibberellins were identified. Understand everyone, among the 150 type of the gibberellins, friends, the first isolated, what my friends, the first isolated and widely used, please remember the statement, the first isolated, good morning Shafia, the first isolated and widely used is the GA3, whereas my friends, the most active form of the gibberellic acid will be the GA1. Please kindly don't forget. Friends, GA1 is the most active form of the gibberellic acid among all. Then friends, ZA3 is the widely studied gibberellic acid and first isolated gibberellic acid. And friends, every one of you listen my statements very carefully. Just now I told to all of you that gibberellic acid induces the stem elongation. That is what I told you. And every student has to remember one important statement. Listen to me very carefully, friends. Gibberellic acid is not going to induce the stem elongation in every single plant. That is not possible. Gibberellic acid is going to induce the stem elongation only in certain plants. Only in the certain plants. Especially what plants? The plants which are dwarf plants or rosette plants. Dwarf plants means which are genetically dwarf. Rosette plants means, friends, every students please try to remember. Rosette plant means, friends, which is growing on the plane of the ground. You can take an example cabbage. You can take an example of radish. You can take an example of broccoli. These are all the one which are basically growing on the plane of the ground. Cabbage, broccoli, radish. Every one of you please kindly look into the diagram. In the first case, you can see, friends, this particular plant is genetically dwarf. By birth, it is a very short plant. But friends, upon injecting the gibberellic acid, what happened, friends? The height was grown. The height was grown. But friends, you only try to look into the third diagram. This particular plant is genetically tall. There is no doubt about it. It is genetically tall. But friends, upon adding the gibberellic acid, do you think is there any increase in the growth of the plant? Not at all. So my friends, what is the statement that I want to conclude here? The statement I want to conclude here is that, listen to me everyone, gibberellic acid always stimulate the stem elongation only in a genetically dwarf plant, which are rosette plants which are growing on the plane of the ground. But what's the statement you have to remember? Gibberellic acid never ever stimulate the elongation in genetically tall plants. Never forget that particular statement. Okay. So friends, I told you, right, what is the best way to remember the hormones, friends? Comparing one hormone with the another hormone. So whenever you are comparing one hormone with another hormone, you can easily remember. So far we have finished only one hormone, that is auxin. I hope all the students remember it. What is the name of the precursor? Friends, focus everyone. What is the name of the precursor that is involved in the biosynthesis of auxin? I hope all of you remember tryptophan. I hope all of you remembered. what is the name of the biosynthetic pathway that is involved in the biosynthesis of oxygen? I hope all of you remember, we called it as the IPA, indole pyruvic acid. Very good. So friends, like that only I want you people to remember. Here, what is the name of the major precursor? Listen once again. What is the name of the major precursor that is involved in the biosynthesis of the terpene? Friends, we will be calling it as the GGPP, zeroanyl diphosphate. What are friends? Zeroanyl biphosphate. How many carbon it is? 20 carbon. It's a terpene. Anyway, we're gonna read it very elaboratively in the secondary metabolites. But I want you people to remember is that GGPP is the major precursor that is involved in the biosynthesis of the terpene. Friends, if you look into the structure of a terpene, if you look into the structure of a gibberellin, you no need to remember the structure at all. Kindly don't worry. But I want you to remember from where it got derived from. Friends, all of you please kindly look into this. This is called as what, friends? Antaquarin. What do you call it as? Antaquarin. What is antaquarin? Antaquarin is a chemical. Exactly, friends. However, the antaquarin looks like, you can see the picture, friends. Gibberellins also looks the same. However, antaquarin looks like, friends, even gibberellins also look same, friends. Please kindly don't forget this. So that's why every student has to listen my statements. In the plant body, if we want to give rise to the gibberellic acid, actually, friends, first we will synthesize the antaquarin. Then, friends, antaquarin is going to give rise to the gibberellins. Please kindly don't forget the statement at all. And friends, I have told to all of you, there are more than, what did I tell you? There are more than 150 type of gibberellic acids are present. I want all of you to remember, my friends, among the 150 type of the gibberellic acid, only few type of gibberellic acids are biologically active. An example, out of 150 gibberellic acid, my friends, just imagine 40 to 50 gibberellic acids are very active. Then friends, what about the remaining? Every student listen to me. Remaining, they can be either in the inactive form as it is, or friends, they are basically acting as a precursor molecule for the activated forms. So all of you listen. Out of 150, only few gibberellic acids are biologically active, my friends. What about the others? Others serve as a precursors, or they will be remain in the inactive form only. Friends, all the hormones are very important. There is a no question paper without the question from the hormone. 4 or 2 marks is in our pocket. That's why you should not miss even a small point also. So friends, listen to me. Among these, some gibberellic acids are having 20 carbon. As I told you, gibberellic acid is derived from the precursor GGPP. So listen, 90% of the gibberellic acids are, friends, they are 20 carbons only. There is no doubt about it. What about the remaining, friends? While others have only 19 carbon, that's all. So remaining carbons can be lost in the form of metabolism. Do not forget the statement. So friends, now look into this. In this particular slide, I am requesting all of you friends, what are the various organelles that are involved in the gibberellic acid biosynthesis? I want you people to remember. You can get a direct question, which of the following organelles are involved in the biosynthesis of the gibberellic acid? Just remember the name of the organelles, my friends, majorly. So it occur in the plastid, endoplasmic reticulum, as well as, friends, in the cytosol. Plastids, ER and cytosol. And friends, what I said to all of you, what is the most active form of the gibberellic acid? I told you the most active form is the GA1. Another one is what? ZA4. And what did I tell to all of you friends? This active form of the gibberellic acid can be derived from what? Precursors. Precursors also gibberellic acids only, right? They will be in the inactive form. What are my friends? They are acting as the precursor forms. Everyone pay attention. Now look into the picture carefully. What did I tell to all of you? If we want to synthesize the gibberellic acid, we require a precursor. What is the precursor we discussed? GGPP, geranyl diphosphate. And my friends, what did I say to all of you? The GGPP is how many carbon containing compound? Friends, I told to all of you the GGPP is a 20 carbon containing compound. Just listen to me carefully. First, friends, this particular GGPP giving rise to the entaquarin, because entaquarin is exactly similar to the gibberellic acid. So friends, first entaquarin can be given by the GGPP, then entaquarin from the plastid, it enter into the endoplasmic reticulum. Then entaquarin give rise to what, friends? ZA12 and ZA53. Now you have to ask me, sir, GA12 and 53 are the most active form of gibberellic acid? Not at all. Then what they are? Friends, they are all acting as the precursors. What, friends? They are all acting as the precursors for the active form of the gibberellic acid. And friends, this particular ZA12 and ZA53 from the endoplasmic reticulum, my friends, they are basically entered into the cytosol. The moment they entered into the cytosol, what is happening, friends? They are going to give rise to the most active form of the gibberellic acids, that is GA1 and GA4. So what's the statement you people has to remember here? The biosynthesis of gibberellic acid actually takes place in the plastid and in the endoplasmic reticulum, as well as, my friends, in the cytosol. So friends, right now what we studied? Just now we understand how the gibberellic acid discovered. We understand a very important statement that gibberellic acid is the one which induced the stem elongation in the dwarf plant or the rosette plants. That is what we studied. Later we understand what are the three organelles that are involved in the biosynthesis of gibberellic acid. Now friends, let's understand the site of biosynthesis and function. Be patient, friends. See, we have to know very depth of every single hormone. So listen the concepts very carefully. I can assure you 100% the question won't go beyond the level that what we are discussing. So but make sure whatever you are learning very depth. Now friends, all of you listen to me carefully. Whenever we studied the auxin, what is the word I have used? Whenever we studied the auxin, I have used a word. Auxin is a rooting hormone. But does the auxin is synthesized in the root? No, right? Auxin is abundantly synthesized in the shoot. They will travel from the shoot to root and they start showing their mechanism in the root. Since auxin is involved in the root development, friends, we can call the auxin as a rooting hormone. Every student has to remember one of the most important statement. The moment you heard the name of a gibberellic acid, one common statement has to strike in your mind. What is that particular statement, friends? This particular gibberellic acid plays a key role in where, my friends? Gibberellic acid plays a key role in the seed germination. Never ever forget the statement at all. And friends, 100% of the, good morning Sumaya, please continue. 100% of the gibberellic acid are found in the immature seeds as well as, friends, developing fruits. Every student just listen to me carefully. Whatever the gibberellic acid that is produced in the root, it will travel to the shoot via xylem. Whatever the gibberellic acid that is produced in the shoot, it travels to the root via phloem. Always remember that statement. But 100% of the gibberellic acids are abundantly found in the immature seeds and the developing fruits, because friends, if the seed germination wants to occur, gibberellic acid should be required compulsory. Friends, now we are going to read every single function most elaboratively. 90% of the question will come either from the function or from the signaling. So friends, it's very important. I want every one of you understand every single function very elaboratively. They are very simple. I'll make it more simple. Just pay attention. Gibberellic acid, it is basically stimulate the stem elongation. How? By stimulating the cell division and by stimulating the elongation. How the gibberellic acid is doing this function, friends, we will read in the next slide very elaboratively in the next slide. Now look into the second function. It breaks seed dormancy. First of all, my friends, all the students remember, what do you mean by the seed dormancy? Everyone listen. Dormancy, dormant is the period where there is a no fertilization. Whenever there is a no fertilization, then there is a no seed germination. Then plants start facing lot of adverse condition. Sometimes plant may die also. Friends, please kindly don't forget, there is a hormone, we're gonna read it. There is a hormone which promotes the seed dormancy. Friends, what is the name of the hormone that promote the seed dormancy, friends? That is the abscisic acid. Every student listen to me very carefully, friends. Whenever abscisic, in the next hormone only we will read, when the abscisic acid promote the seed dormancy, there will be a no fertilization. Friends, whenever there is a no fertilization, there is a no seed germination, then the plant has to face lot of adverse condition like drying, desiccation. What is desiccation? Desiccation is the condition where there is absolutely no water content in the plant body. Plant may die, right? So abscisic acid promoting the seed dormancy. But my friends, when you look into the gibberellic acid, friends, gibberellic acid is going to break the seed dormancy, which means, friends, it is just performing the opposite function to the abscisic acid. Everyone listen to me carefully. When abscisic acid promote the seed dormancy, no fertilization, no seed germination. But friends, when the gibberellic acid break the seed dormancy, friends, there is a fertilization, there is a seed germination. So my friends, I can conclude one statement to all of you. What is the statement that I can conclude? I can conclude a statement that abscisic acid and gibberellic acid both are antagonistic to each other. My friends, what do you mean by the word antagonistic to each other, which are acting opposite to each other? I hope every one of you remember, while we are studying the auxin, I told you very clearly to all of you, auxin and cytokinins are opposite to each other or antagonistic to each other in apical dominance. I hope you remember. Auxin promote the apical dominance, cytokinin inhibit the apical dominance. I hope all of you remember, my friends. Same way here also, when you look into the gibberellic acid break down the seed dormancy, abscisic acid promote the seed dormancy. Always keep that particular point in your mind. Then the third and very unique point. From this point, so far we got 3 part B questions. Direct 3 part B questions we got from this particular point. I will explain this point in the upcoming slides very elaboratively by using the diagram. But listen, gibberellic acid helpful in what, friends? Production of the alpha amylase. Friends, what is alpha amylase, friends? Alpha amylase is what? Alpha amylase is the hydrolytic enzyme. Alpha amylase is what? Hydrolytic enzyme. What is the function of alpha amylase? Breaking down the starch into simple sugars like glucose. So you can get a direct part B question also. Which of the following hormone is responsible for the stimulation of the alpha amylase? Obviously, my friends, that is the gibberellic acid only. Next point, friends, the gibberellic acid stimulates flowering in response to long days. Friends, please kindly don't confuse with this particular long day plant, short day plant. We are going to read in this sensory photobiology chapter very elaboratively. But for all of you, I'm just telling you right now, when you can called a plant is the long day plant, when you can called a plant is the short day plant. See friends, any plant, everyone listen, any plant, if it wants to flower, it require a day length. We can call, my friends, critical day length. What do you call, friends? Critical day length. How much it is? 10 to 12 hours. So every student, please listen to me carefully. Any plant, if it wants to flower, it requires minimum 10 to 12 hours of sunlight. If any plant, in order to flower, if it is using less than 10 to 12 hours, we will be calling it as the short day plant. If any plant, in order to flower, it is using more than 10 to 12 hours, friends, during that time we will be calling it as the long day plants. Anyway, that we are going to read in the sensory photobiology, don't worry at all. Now what I want you to remember here, whenever there is a presence of the gibberellic acid, you know what it will do? Gibberellic acid plays a key role in stimulating the flowering, mostly in the long day plants when compared to the short day plants, which means gibberellic acid stimulate the flowering in the plants which are using the sunlight more than 12 to 14 hours in order to flower. That is what do we call stimulating the flowering. And another very important statement, friends, gibberellic acid induces maleness in dioecious flower. I hope, my friends, every one of you remembered this particular point. What is the name of the hormone that induced the femaleness? I hope you remember. In case of a dioecious flower, auxin is the hormone which induces the femaleness in dioecious flower. Once again, I am reminding to all of you, never ever confused. Dioecious means not two. Dioecious means either male or female. So which means, my friends, dioecious means monosexual. Good, auxin. Dioecious means what, friends? Monosexual. So friends, whenever there is a presence of the gibberellic acid most of the time it induces the maleness when compared to the femaleness in the dioecious flower. Please kindly don't forget the point. Good. Next, another important function, friends, it causes parthenocarpic fruit development. My friends, what do you mean by the word parthenocarpy? Everyone listen to me. Parthenocarpy means what, friends? Seedless fruits without fertilization. I hope every student already remembered what is the name of the hormone that is involved in the parthenocarpy. We studied, friends, auxin. Auxin is the hormone which is involved in the parthenocarpy. Along with the auxin, my friends, even gibberellic acid is also involved in the parthenocarpy. And friends, this statement is a kind of contradictory statement. It delays the senescence in the leaves and citrus fruits. First of all, every student has to answer me. What is senescence, friends? Becoming old, aging. Now, every student listen my statements very carefully and don't get confused. Suppose example, in the exam, they have asked us a question. Which of the following hormone is involved, or which hormone enhances senescence? Enhances senescence means Which hormone is responsible for aging of a leaf and fruit? All the time you have to remember an answer as ethylene. Never forget that. If they ask you a statement, which of the hormone, I will ask you in the next hormones also, I want you to remember, friends. If they ask you a question in the exam, what is the name of the hormone that is involved in delaying the senescence? My friends, every student has to remember the original answer for this is the cytokinin. I am requesting all of you friends, please kindly don't forget these two important statements. Hormone ethylene is allowing the leaf to become older, allowing the fruit to become older. By that they'll fall. But when the cytokinin is present in the plant, it never allow the leaf and fruit to become older very faster. So it delay the senescence. Now you can ask me, sir, then why you are talking about the gibberellic acid? Listen friends, gibberellic acid is also involved in delaying the senescence. There is no doubt about it, but not in all the cases. Especially gibberellic acid delay the senescence, my friends, in the leaves and citrus fruits only. Don't forget this particular statement. But friends, major hormone you should use the word cytokinin only. Don't forget that. Okay, these are all the various functions of the gibberellic acid, my friends. All these functions we are going to read elaboratively, which is extremely important for exam. Don't forget even a single function also. Now let's talk about the first function. How gibberellic acid is involved in the stem elongation. Friends, from this particular slide till the end of the signaling, it is little confusing. I am requesting every student please focus 20 minutes continuously without interruption. Yes, Sadhguru, seed dormancy also plays a key role in the parthenocarpy. Yes, but it did not have the major role. Did not have the major role. I can use a word, it is having the partial role in the seed dormancy. Sorry, partial role in the parthenocarpy. Okay, okay. Friends, listen to me carefully from this particular slide. How gibberellic acid is induced at the stem elongation. I want every student has to remember one interesting point. What is that? I have already conveyed, or I already told you that gibberellic acid is basically inducing the stem elongation, friends, especially in the dwarf plants. Where friends, especially in the dwarf plants or the rosette plants. I hope all of you remember, my friends, what is a rosette plant? I told to all of you, rosette plants are the plants which are growing on the plane of the ground. So gibberellic acid is not at all involved in the stem elongation in every single plant. And one more very interesting point, you have to recall the auxin whatever we discussed. Gibberellic acid never ever involved in the stem elongation without auxin. That's a really an interesting statement. So listen, gibberellic acid always depends on what? Understand, use the brain. Understand for the next 20 minutes correctly. Gibberellic acid depends on the auxin induced cell wall acidification, which means, my friends, listen to me carefully. If the gibberellic acid wants to involve in the cell elongation, it is compulsory auxin has to present in the cell, in the plant. If there is a no auxin, gibberellic acid never ever elongate the cell wall. That's a common statement. That's why we have written gibberellic acid is never present in the tissues in the absence of auxin. I am requesting all of you quickly recall in the last class, day before yesterday class, we explained very clearly for the period of 10 minutes how the auxin is going to induce the cell wall elongation. Suppose this is the cell membrane, the same diagram we read, and cell wall and cell membrane. We read that on the cell membrane what kind of pumps are present? H plus ATPase pumps are present. I hope every student remembered. Now auxin hormone will come and activate the H plus ATPase pump. H plus ATPase pump pumps the proton. Where the proton pumps the proton? Into the cell wall space. Cell wall space become acidic. I hope every student remember, in the acidic environment, what protein is getting activated? Expansin is getting activated. Now I hope I have drawn the same diagram and I explain it to you. What is the function of expansin? I told to all of you, expansin is the one which can loosen the cellulose microfibrils. I have explained you like this. Listen to me carefully. Cellulose microfibrils are like this. They are associated very tightly with each other. What did I tell to all of you? This expansion will go and loosen the cellulose microfibrils like this. Once they loosen the cellulose microfibrils like this, more amount of the water enter inside. The cellulose microfibrils become turgid. Cell wall get elongated. That's a simple point. Who is doing that? Who is entering into the cellulose microfibrils? Expansin enter. Now every student has to listen. How does the expansin enter? Who will incorporate the expansin into the cellulose microfibril? Everyone listen my statements. Who will incorporate the expansin into the cellulose microfibril? My friends, this can be mediated by an important protein called as XECT protein. Very simple, my friends. XECT is the name of the protein which will allow the expansin, which will incorporate, which will penetrate the expansin into the cellulose microfibrils. Now the question comes here, how this particular XECT protein is getting activated, my friends? Friends, the XECT protein is getting activated because of gibberellic acid. That's all. Very simple. So gibberellic acid function is only what? Activating the XECT protein. Activated XECT protein activate the expansin. Activated expansin penetrate into the cellulose microfibril, loosen the cellulose microfibril, allow the water movement, obviously cell get elongated. Very simple point. So friends, what is the statement that I can conclude to all of you? Gibberellic acid always show the cell elongation with the help of auxin. And what is the role of the gibberellic acid here, my friends? Gibberellic acid is the one which is involved in the inducing this particular protein XECT. What the XECT is doing, my friends? The hormone XECT facilitate the penetration of the expansin into the cell wall. Never ever forget this. And what is the most important statement? Gibberellic acid stimulate the cell elongation by using XECT protein also and expansin protein also. Very simple statement it is. Now we understand how the gibberellic acid is involved in the cell elongation. Friends, let's try to understand, focus, how the gibberellic acid is going to involved in the cell division. Friends, all of you pay attention. It is very simple. Here you have to remember, gibberellic acid actually plays a key role in the cell division by activating or by stimulating several CDK molecule. Because we read in the unit 2 very clearly, how does the cell cycle can be controlled, friends? Regulated with the help of CDK protein, with the help of cyclin proteins only. We all know that. Now the question comes here, how does this particular gibberellic acid is inducing the transcription? It is inducing the transcription by stimulating or by activating the CDKs. How is that possible? Friends, gibberellic acid stimulating the CDKs by activating the CDKs which are involved in the G1-2S phase. It will also activate the CDKs which are involved in the G2-2M phase. In the unit 2 we read which CDKs are involved in the G1-2S phase, and we also read, my friends, which CDKs are involved in the G2-2M phase also. So friends, right now what we understand is the functions. there is another important function that I want to highlight. I told you that is this function, that is gibberellic acid stimulates. What it's going to stimulate? It is going to stimulate alpha amylase in the germinating cereal grains. My friends, all the students, listen to me. From this particular diagram, we got three times, sorry, not three times, two times direct part B question. One time as one of the statement, not complete question, as one of the statement in the part C. Now all of you listen my statements very, very carefully. What I am explaining to all of you, listen to me 5 minutes this particular diagram carefully. Here in this diagram we are explaining actually how the gibberellic acid is involved in the stimulation of this particular alpha amylase. Every student listen to me. What is the function of alpha amylase? It is a hydrolytic enzyme. We all know very well. Its function is what? It is going to break down the starch into glucose. By that the embryo can use this. Now friends, all the students look into the diagram carefully. This is the embryo. You can see, right, that this one, this entire thing we called as starchy endosperm. My friends, why did I use a word starchy endosperm, friends? Because you can see right inside it's completely white, cream color or white color. That is the only starch. Now every student listen to me. This is the embryo. All of you understand what exactly I am saying. If the embryo wants to grow, if the embryo wants to develop, what the embryo require? Embryo require the food. Food in the form of what? Glucose. Listen to me carefully. Whatever, if the embryo wants to grow and if the embryo wants to develop, it requires the food in the form of a glucose. But the point is here, my friends, this particular embryo cannot break down this particular starch directly. Now what is happening? Every student try to understand. Listen to me carefully, I'll explain twice. Whatever the gibberellic acid that is present in the embryo, that gibberellic acid from the embryo, where it is reached, my friends, from the embryo it is reached into the endosperm. From the embryo it reached into the endosperm. Now the question is here, how the gibberellic acid reached from the embryo into the endosperm? Via a structure called as scutellum. This is the first time asked part B question. Which of the following structure will help the gibberellic acid to enter from embryo into the starchy endosperm? That is the scutellum. Okay, finally it entered into the endosperm. Now what is going to happen? Now this particular gibberellic acid, my friends, it is diffuses into a particular layers of the starchy endosperm. What is that layers we called as? We called aileron layers or aileron cells. This is the second time asked part B question. In which of the following layers gibberellic acid can undergo the diffusion? That is the aileron layer. My friends, when the gibberellic acid diffuses into the aileron layer, immediately what is going to happen? Signaling will occur. Then finally, I will tell you the signaling in the next slide. Aileron layers signaling will occur. Finally, they produce what, my friends? They produce abundant amount of the hydrolytic enzymes. Friends, among all the hydrolytic enzyme, what is the most important hydrolytic enzyme? That is only alpha amylase. Now the alpha amylase break down the starch, convert into glucose. Now the question is very clear. Now how does the glucose enter from the endosperm into the embryo? Because the embryo is the one who require the glucose right now. Now it is going via this particular scutellum only, and it reaches to the embryo as well as it reaches to the root also. Whomever did not understand, my friends, I am repeating it once again. Please try to understand me. Gibberellic acid, how it is inducing the alpha amylase in the embryo. Everyone try to understand. Whatever the gibberellins that are synthesized by the embryo, they are released into the starchy endosperm via scutellum. Once the gibberellic acid entered into the starchy endosperm, gibberellic acid will directly diffuse into the aileron layers of the starchy endosperm. On the aileron layer, certain signaling will happen. We will see that. Then finally it will synthesize the proteins or enzymes called as hydrolytic enzymes. One of the major hydrolytic enzyme is what, friends? Alpha amylase. That alpha amylase enzyme break down the starch into simple sugars like glucose. Now that particular simple sugars, friends, they will travel once again via scutellum. They enter into the embryo and root. Then my friends, then the embryo and root starts using it. Simple. This is how gibberellic acid induces alpha amylase in embryo. If the question is difficult, we are going to get the question from the signaling. Now I will explain the signaling very elaboratively to all of you. Later I will solve all your doubts. Focus, pay utmost attention. Gibberellic acid signaling. How does this particular signaling will happen, sir? What are the various proteins that are involved in the signaling? My friends, it is very important. It is mandatory. You people has to remember the signaling compulsory, because if the question comes very tough in terms of plants, plant hormones I'm talking, we will get the question from the signaling only. Every student pay attention. Gibberellic acid is going to regulate various plant developmental processes in the plant. So listen everyone. Gibberellic acid is required for seed germination. Very, very important. It required for seed development. That's also true. And friends, it is also required for the flower induction. We read that. And friends, every student has to remember one of the most important point here. How does this particular gibberellic acid signaling will be controlled? Friends, the gibberellic acid signaling will be controlled by an important protein. What do you call that particular protein, friends? We called that particular protein as the DELA protein. The moment you heard the name of a DELA protein, one common statement you have to remember, that it is present in the nucleus. This is the statement you have to remember. In the given exam, in a 4-marker question, they have already asked this point. DELA protein is a positive and negative regulator of the GA present in the nucleus. Of course, true point it is. Keep it in your mind. DELA proteins are the class of nuclear proteins that act as a transcriptional regulators and suppressors for the gibberellic acid signaling. Don't forget the point. Friends, listen to me. So far, 5 DELA genes were identified in the Arabidopsis thaliana, which is a jasmine-like seedling. It's not jasmine. Jasmine-like seedling. Vorija sativa, in the rice, there will be only 1 DELA gene was identified, and this statement was asked in the exam 2019 June exam as one of the statement. Now you know very well. Please remember, friends, I am requesting every one of you to remember this particular 5 DELA genes. It's difficult to remember, but you have to remember. There's no shortcut here. At least by chance, if you can't remember this 5, at least I am requesting all of you remember, in Arabidopsis thaliana there are more than 5 DELA genes were identified. Remember that statement. And in Vorija sativa, remember, so far there is only 1 DELA gene were identified. And friends, I hope every student remember. I am not asking you the questions. Whenever we are solving the question paper, then you have to answer every time. Auxin, I hope you remember, it will associated with which kind of a receptor? We called tier 1 receptor. The question which was asked in the 2020 November paper. Along with that, they have also given what? Gibberellic acid. Gibberellic acid is associated with which receptor, my friends? GID1 receptor. The same question. So I am requesting all of you, remember, friends, Gibberellic acid associated with a receptor which we call, my friends, GID1. Don't forget it. Once again, when I am talking about the other hormones, I will compare, then you can understand whether you remember the concept or not. Okay, so friends, every student try to understand how does this particular gibberellic acid signaling will takes place, and all the students listen my statements carefully. Suppose example, listen, if you recall this particular 2 points, the concept is very clear to all of you. If there I already told to you, right? If you want to remember the signaling pathway of the any hormone, the best shortcut is you have to understand what happens in the plant body in the absence of hormone. I will explain you what happens in the plant body in the presence of hormone. Simple point. If you remember those two, the concept is very clear. Now every student try to understand, whenever there is a absence of gibberellic acid, my friends, who is going to get activated? Della protein get activated. Della protein get activated. My friends, della protein is a suppressor, right? When the della protein get activated, my friends, there is a no expression. It leads to the repression. Means gibberellic acid is never ever get expressed because della repressors are active. My friends, now what's the second condition? Whenever gibberellic acid is present, what the gibberellic acid will do? It will come and associated with the GID1 receptor. It's a type of G protein only. Now, once the GID1 receptor, gibberellic acid associated with the GID1 receptor, della protein get degraded. Then the gibberellic acid expression will occur. Remember these two statements enough. I will make the signaling extremely simple. I am requesting all of you look into the diagram very carefully. I will explain twice elaboratively. Focus. Let's talk about this, friends. In the presence of the gibberellic acid, every one of you listen to me carefully. So friends, this is the gibberellic acid receptor. All the students remember. And to this receptor, who will come and attaches, friends? Gibberellic acid 1 will come and attaches. The moment gibberellic acid 1 will come and attaches, immediately what is happening? There will be a activation of the GPCR and the G protein. Immediately what is happening, friends? Every student listen to me carefully. The gibberellic acid signaling pathway occur in two ways. One we call it as what, friends? Calcium independent pathway and calcium calmodulin dependent pathway. I'm sorry. One second, friends. I'm so sorry. Yeah. So what I am saying, the gibberellic acid signaling will occur in two ways: calcium dependent and calcium independent. Okay. Everyone look into the picture carefully, friends. Whenever gibberellic acid come and associated with its receptor, it activates the G protein. Now the calcium dependent and independent pathway will start. Now let's talk about the calcium independent pathway. I'll explain twice, listen. Now this activated gibberellic acid signaling molecule, all of you look into this picture, it is entering into the nucleus. Friends, the moment gibberellic acid enter into the nucleus, every student has to listen to me carefully. This is what gene present in the nucleus? Alpha amylase gene. Just now we studied very clearly, right? This particular alpha amylase gene has to be activated. Then only we can produce alpha amylase. Now listen, but everyone understand, do you think alpha amylase gene is really free? No, right? Alpha amylase gene is actually blocked. Who blocked this particular alpha amylase gene, friends? This alpha amylase gene is blocked by the della repressor. So what's happening now? The gibberellic acid enter inside. Already della repressor blocking the alpha amylase gene. So gibberellic acid associated with the della repressor. Now my friends, della repressor come out. Then the della repressor can be degraded by 26S proteasomal complex. I'll explain twice, just listen. Now which gene become free, friends? Alpha amylase gene become free. Now the gene undergo the transcription, translation. Finally, what does it produce? It produces the alpha amylase protein or our enzyme. Now what's happening now? That alpha amylase packed in the endoplasmic reticulum and associated with the Golgi complex undergo the modification in the Golgi complex. Finally, friends, they packed in the form of a vesicle, and that vesicle, the vesicle contains what? The vesicle contains alpha amylase. Now, how does that particular vesicle can come and fuse the membrane? That's the point. That is in the presence of calcium. Calcium will allow this particular vesicle to fuse the membrane. Once they fuse the membrane, alpha amylase released. What alpha amylase do? Break down the starch into glucose. Then via scutellum, glucose enter into the embryo and root. They will use it. So friends, all of you listen. Where this particular signaling is happening, every student has to look into the diagram. This signaling is happening. This is the aleurone cell, right? Exactly on this membrane, this particular signaling is happening. So this is what membrane? This is the aleurone layer membrane, friends. So look into the picture carefully once again, every one of you. Whenever the gibberellic acid come and associated with the gibberellic acid receptor, it activates the G protein. Then there are two kinds of pathways will occur. One we called calcium independent pathway, another one we called calcium dependent pathway. Whenever there is a activation of the in case of calcium independent, do not require calcium, the gibberellic acid signal get activated. The activated gibberellic acid signal will enter into the nucleus, and here alpha amylase gene is already blocked. Now this alpha amylase gene is associated with what? Delta repressor. That's why it got blocked. Now gibberellic acid will associated with the delta repressor. Delta repressor immediately get degraded by a degradative complex. What was that? 26S proteasomal complex. Then alpha amylase gene undergo the transcription, translation. Finally, it produce the alpha amylase, and this particular alpha amylase packed in the Golgi complex, it undergo the modification. Finally, my friends, that vesicle in the presence of calcium will go and fuse the membrane. They release the alpha amylase. Alpha amylase break down the starch into glucose. Finally, glucose enter into the embryo. Then embryo can use it. That is gibberellic acid signaling pathway, and the entire hormone is completed. Now you can ask all your doubts. Okay. Friends, say yes or no, everyone. Is that clear, everyone? The hormone gibberellins. Is it clear, everyone? Please say yes or no. You can ask me any kind of a doubts, whatever you have. So friends, in maximum 7 days. Okay. So today what is the date? I am telling you upcoming plan. Every one of you listen. Today is 22nd, right? So maximum by 30th. Maximum by 30th, friends. The entire chapter will be completed. Which chapter? Unit 3 will be completed. Unit 6 also will be completed. Deepika, I will explain you the last step. Ask me, Deepika, what step you need exactly, please ask me. So Unit 3 will be completed and Unit 6 also will be completed. Okay. And what I will do on 31st March, just make this particular date. See, 31st night, most probably in the evening session. That is 8:30 to 11:30. How much time it take doesn't matter. In the only night time, we will finish all the PYQs of the Unit 3 and Unit 6. At least 5 question papers. By that you will get enough of confidence to answer all the questions. This is my goal. And friends, from the April 1st, in the endosperm, Sumaya, in the endosperm. Okay, in the April, I am requesting every student right now. I am telling you, informing you right now. I will take total 3 hours of classes in the morning. Okay, one class I will take here to all of you 5:30 to 6:30, and immediately I will take you 7 to 8 a.m. and I will take another class 8:30 to 9:30 a.m. Okay, These two I will be taking in the Unacademy. You can join after immediately. So my goal in the month of April is finishing minimum 3 to 4 units. I'll finish it for very sure, undoubtedly. So we have to work this time very vigorously, friends. By that we can achieve our target very faster. Okay. Please make sure that I don't expect anything from all of you. Just be consistent. Just work hard with more interest. Enough. And Sumaya, you can look into this. The white color you can see, right? That is the endosperm. In that endosperm only, starch can break down into glucose. And the glucose, I am showing the arrow. It enter into the embryo. And you look into this particular picture, Deepika. Once the vesicle get formed, see here, the alpha amylase protein got synthesized. It is packed in the form of a vesicle. From the ER, that vesicle come to Golgi complex. From the Golgi complex, the vesicle come out, and that vesicle can fuse the membrane. Whenever it is fusing the membrane, it require compulsory calcium. So in the presence of calcium, the vesicle fuse the membrane, release the alpha amylase. You got it? Yes, Sadguri, all in Unacademy, all the free classes. Whatever it may be, friends, I will finish the syllabus on right time, not on right time, before the time. And we will put the more effort in the revision and question solving. Okay, friends, that's all for today. Okay, we'll meet in the tomorrow morning class. Thank you so much. Please kindly revise it. Okay, please revise, kindly don't waste the time. I will see you in the tomorrow morning 5:30 a.m. Bye-bye. So tomorrow we are going to do the left part of the RNA processing and translation. Thank you so much. Bye-bye. Good night to all.